A very good day I greeted to Madam Shazila binti Snavi, our respected lecturer of the subject Microbiology. So today our group G is going to present about Saccharomyces cerevisiae in food production. So this is my group members, myself Lourdes of Kalimara, number Matrix 22 21F 2034, and my other group member is Lagishni Naidu Anapapasegre, number Matrix 23 21F 2022, and another group member is Dishani Professor Kumar, number Matrix 23 21F 2042. So next will be the introduction part. So Saccharomyces cerevisiae is commonly known as a baker yeast or a brewer yeast. It is also a species of a yeast that is commonly found in a natural various habitat. So here are some examples of a natural various habitat. So firstly will be the fruits and vegetables, second will be the soil, third will be tree and plants, and fourth will be the wild fermentation. So firstly, let's look on the fruits and vegetables. Saccharomyces cerevisiae is often found in the skin of various fruits such as grape, berries, and apples. It also can be found in the surface of some vegetables such as cucumbers. The next will be the soil. These yeast species can be found in soil, particularly in areas where fruits and vegetables are grown. It is often present in upper layer of the soil where it can ferment plant material or organic matter. The next will be the tree and plants. Saccharomyces cerevisiae can inhabit the bark and leaves of the trees and various plants. It can be also found in natural environment where it can ferment the sugar present in the sap or exudates of these plants. So next will be the wild fermentation. Saccharomyces cerevisiae is also associated with natural fermentation processes. It can be found in the wild of surfaces of plant materials such as fallen fruits or flowers, where it initiates the fermentation process by converting sugars into alcohol and carbon dioxide. It is important to note that Saccharomyces cerevisiae can be found in various natural habitats. It has also been extensively domesticated and used for commercial purposes in the production of bread, beer, and wine. In these industrial settings, the yeast is cultivated under controlled conditions to optimize its growth and the fermentation capabilities. The next part will be the specialized characteristics of Saccharomyces cerevisiae based on their metabolism. Saccharomyces cerevisiae possess several specialized characteristics based on its metabolism, which contributes to its widespread industrial use and research interests. Here are some key characteristics. So firstly will be the fermentation process. Saccharomyces cerevisiae is a facultative anaerobes. They carry out both aerobic respiration and fermentation process in the absence of oxygen. This yeast is known for its ability to efficiently ferment sugar such as glucose and fructose into alcohol, which is ethanol, and carbon dioxide through a process known as alcoholic fermentation. This characteristic is exploited in the production of beverages production such as beer and wine, as well as in the industrial ethanol production. Next will be the cap-tree effect. Saccharomyces cerevisiae exhibits the cap-tree effect, which refers in the preference for the fermentation even in the presence of sufficient oxygen. When the glucose levels are high, Saccharomyces cerevisiae represents respiration and prefer to carry out fermentation process. This metabolic feature is advantageous in industrial application where high ethanol production is desired, such as in brewing and biofuel production. Next will be utilization of diverse carbon source. Saccharomyces cerevisiae is capable of utilizing a wide range of carbon sources beyond glucose. It can metabolize other sugars such as fructose, maltose, and sucrose, as well as non-sugar carbon sources like organic acids such as citric acid and alcohol such as glycerol. This versatility makes it valuable in various industrial processes including the production of biofuels, organic acid, and other metabolites. Tolerance to stress condition will be the next point. Saccharomyces cerevisiae has developed mechanism to tolerate various stress conditions. It can withstand high osmotic pressure, acid or alkaline pH, and elevated temperatures. This stress tolerance is crucial in industrial fermentation, where the yeast is exposed to harsh conditions, enabling it to survive and continue fermentation under challenging conditions. Next will be the nutrient assimilation. Saccharomyces cerevisiae has a robust nutrients assimilation system, allowing it to efficiently take up and utilize essential nutrients for growth and metabolism. It requires nitrogen sources such as amino acid and ammonia, vitamins and minerals for optimal growth. Understanding and manipulating its nutrient requirements have practically applications in industrial biotechnology.
Nutritional type based on their carbon and energy source. Saccharomyces cerevisiae is a heterotrophic organism, meaning it obtains its carbon and energy from organic source. More specifically, it is classified as facultative anaerobic fermentative yeast. First, carbon source. Saccharomyces cerevisiae is capable of utilizing various carbon sources for its growth and metabolism. The primary carbon source for yeast is glucose, which ferments into ethanol and carbon dioxide. However, it can also metabolize other sugars such as fructose, maltose, sucrose and galactose, as well as non-sugar carbon sources like organic acids and alcohols. Next is energy source. Saccharomyces cerevisiae derives its energy through the process of fermentation. During fermentation, it converts sugar into energy-rich molecules, primarily in the form of adenosine triphosphate (ATP), without the requirement of oxygen. However, under aerobic condition, it can also carry out aerobic respiration and generate ATP through the complete oxidation of glucose. Overall, Saccharomyces cerevisiae is metabolically versatile and can adapt to a wide range of carbon and energy sources, allowing it to thrive the various natural and industrial environment. In its natural habitat, which is obtain essential nutrients like nitrogen, phosphate and potassium from its surrounding. Nitrogen. Saccharomyces cerevisiae requires nitrogen for protein synthesis, nucleotide synthesis and other essential cellular processes. In its natural habitat, nitrogen sources are commonly found in the form of amino acid. This nitrogen source can be present in soil, organic matter or decaying plant material. It possesses specific transporter on its cell membrane that enables it to uptake and utilize nitrogen compound present in its environment. Next is phosphate. Phosphate is crucial for Saccharomyces cerevisiae as it involves in DNA and RNA synthesis, energy metabolism via ATP and cell membrane synthesis. In its natural habitat, phosphate is found in the soil and organic matter, primarily in the form of inorganic phosphate ions. It possesses transports that facilitate the uptake of phosphate from its environment, allowing it to satisfy its phosphorus requirements. Next is potassium. Potassium is an essential mineral required for maintaining osmotic balance and enzymic function and cellular metabolism in Saccharomyces cerevisiae. In its natural habitat, potassium ions are commonly found in the soil and water. The yeast can uptake potassium through specific transporter in its cell membrane, ensuring an adequate supply of this mineral for its physiological needs. In its natural habitat, Saccharomyces cerevisiae can scavenge and utilize nitrogen, phosphate and potassium from its surrounding environment to support their growth and metabolic activities. The availability and concentration of these nutrients can influence the yeast growth rate, fermentation efficiency and overall fitness in its natural ecosystem. Next is oxygen requirement. Saccharomyces cerevisiae is a facultative anaerobe. It can carry out both aerobic and anaerobic metabolism, which depends on the availability of oxygen. Capability to grow and survive in the presence of and absence of oxygen, exhibiting flexibility in the metabolic pathways. There is aerobic metabolism and anaerobic fermentation. Saccharomyces cerevisiae displays the ability to adapt its metabolic pathway based on the oxygen availability and its environment. Aerobic metabolism. In the presence of oxygen, Saccharomyces cerevisiae can perform aerobic respiration, which involves the complete oxidation of glucose to carbon dioxide and water. Next, anaerobic fermentation. In the absence of oxygen, Saccharomyces cerevisiae is capable of carrying out anaerobic fermentation. It metabolizes glucose through the process of alcoholic fermentation, converting it to ethanol and carbon dioxide.
How they neutralize toxic byproducts of oxygen if they utilize oxygen. When Saccharomyces cerevisiae utilizes oxygen during aerobic metabolism, it faces the potential production of toxic byproducts known as reactive oxygen species ROS. ROS are highly reactive molecules including superoxide radicals, hydrogen peroxide, and hydroxyl radicals, which can cause damage to cellular components such as proteins, lipids, and DNA. To neutralize these toxic byproducts, Saccharomyces cerevisiae employs various defense mechanisms, including antioxidant enzymes, glutathione, DNA repair mechanisms, heat shock proteins, and regulation of antioxidant gene expression. Antioxidant enzymes. Saccharomyces cerevisiae produces antioxidant enzymes that act as scavengers of ROS. These enzymes include superoxide dismutase, also known as SOD, catalase, also known as CAT, and glutathione peroxidase, which is known as GPX. SOD converts superoxide radicals into hydrogen peroxide, while catalase and GPX break down hydrogen peroxide into water and oxygen. By rapidly removing ROS, these antioxidant enzymes help protect the yeast from oxidative damage. Next is glutathione. Glutathione is a tripeptide which is composed of glutamate, cytosine, and glycine that serves as an important antioxidant molecule in Saccharomyces cerevisiae. It acts as a cofactor for various enzymes involved in detoxification processes and also directly scavenges ROS. Glutathione helps maintain the cellular redox balance and protects important cellular components from oxidative damage. Next is DNA repair mechanisms. Saccharomyces cerevisiae possesses DNA repair mechanisms that are specifically activated in response to oxidative stress. These mechanisms include base excision repair, which is BER, nucleotide excision repair, which is NER, and homologous recombination repair, which is HRR. These repair pathways help correct DNA damage caused by ROS and maintain genomic stability. Heat shock proteins. Heat shock proteins, HPS, are molecular chaperones that play a crucial role in protein folding and protecting proteins from oxidative damage. Saccharomyces cerevisiae induces the expression of HSPs under conditions of oxidative stress to prevent the aggregation and misfolding of proteins caused by ROS. And lastly, regulation of antioxidant gene expression. Saccharomyces cerevisiae has a complex network of transcription factors and signaling pathways that regulate the expression of antioxidant genes in response to oxidative stress. One such transcription factor is YAP1, which activates the transcription of genes involved in antioxidant defense when exposed to ROS. By employing these defense mechanisms, Saccharomyces cerevisiae can effectively neutralize the toxic byproducts of oxygen and minimize oxidative damage to maintain cellular integrity and function during aerobic metabolism. pH requirement. Saccharomyces cerevisiae has a pH requirement for optimal growth and metabolic activity. While it can tolerate a range of pH values, it exhibits the highest growth rate and fermentation efficiency within a specific pH range. Saccharomyces cerevisiae generally prefers a slightly acidic to neutral pH range for optimal growth. The pH range that is most suitable for its growth and fermentation varies depending on the specific strain and the application. However, a pH range of 4.0 to 6.0 is commonly considered favorable for Saccharomyces cerevisiae. Within the preferred pH range, Saccharomyces cerevisiae typically exhibits an optimum pH for its growth and metabolic activity. The optimum pH for Saccharomyces cerevisiae growth is usually around pH 5.0. This pH value promotes efficient nutrient uptake, enzymatic activity, and cell proliferation. It's important to note that Saccharomyces cerevisiae exhibits some strain-to-strain variation in pH tolerance and optimum pH. Certain strains may be more tolerant of acidic or alkaline conditions, and the optimum pH may deviate slightly from the general range mentioned above. In various industrial applications such as brewing, wine making, and baking, the pH of the fermentation or dough is carefully controlled to maintain an environment that is conducive to the growth and fermentation of Saccharomyces cerevisiae. This helps ensure optimal yeast performance and the desired product quality. Temperature requirement. Saccharomyces cerevisiae has specific temperature requirements for its growth and metabolic activity. The temperature requirement type of Saccharomyces cerevisiae is classified as mesophilic. Saccharomyces cerevisiae thrives within a relatively narrow temperature range and is considered a mesophilic yeast. It prefers moderate temperatures for optimal growth and fermentation. The optimum temperature for Saccharomyces cerevisiae growth and metabolic activity typically falls between 25 degrees Celsius to 30 degrees Celsius. Within this temperature range, the yeast exhibits the highest growth rate and fermentation efficiency. While Saccharomyces cerevisiae prefers moderate temperatures, it can tolerate a broader temperature range to some extent. The lower temperature limit for growth is typically around 4 degrees Celsius, while the upper temperature limit is generally around 40 degrees Celsius. However, it's important to note that the yeast's growth rate and fermentation efficiency decreases significantly outside its optimal temperature range. Controlled temperature plays a crucial role in industrial processes where Saccharomyces cerevisiae is utilized, such as brewing, wine fermentation, and bread making. Maintaining the appropriate temperature within the optimal range ensures the yeast's optimal performance and the desired product quality.
So next will be the type of industry that are being used. Saccharomyces cerevisiae has widespread use in several industries due to its fermentation capability, metabolic versatility, and ease to cultivation. Here are some notable industries where Saccharomyces cerevisiae is commonly utilized. Firstly will be the food and beverage industry. Saccharomyces cerevisiae is extensively used in food and beverage industry, and it plays a vital role in the production of bread, where it ferments sugar in the dough to produce carbon dioxide in order to rise the bread. In addition, Saccharomyces cerevisiae is a key ingredient in the fermentation of processes of beer and wine and spirits. It converts sugar into alcohol and carbon dioxide, imparting characteristic flavors and alcohol contents to these beverages. So next will be the biotechnology and biofuel production. Saccharomyces cerevisiae are widely employed in biotechnology applications, including the production of biofuels. It is used to ferment sugar that derives from biomass feedstocks such as corn, sugarcane, and lignocellulosic material into bioethanol, which serve as a renewable fuel source. This yeast is also utilized in the production of valuable bio-based products such as organic acid, which is citric acid, and enzymes. Next will be the pharmaceutical and nutraceutical industry. Saccharomyces cerevisiae is utilized in the production of various pharmaceuticals and nutraceuticals. It serves as host organism for the production of therapeutic protein, and including vaccines, antibodies, and enzymes. This yeast's ability to produce and secrete proteins make it a valuable platform for biopharmaceutical manufacturing. In addition, Saccharomyces cerevisiae is used as probiotic and nutritional supplement due to its beneficial effects on gut health and nutrient assimilation. So next will be scientific research and genetic engineering. Saccharomyces cerevisiae have long been a model organism for scientific research, where it is well understood genetic, easy of manipulation, and fast growth make it excellent tool for studying fundamental cellular processes, genetic, and microbiology. The yeast genetic engineering capabilities have enabled the production of strain with enhanced fermentation process properties, stress tolerance, and specific metabolic pathways for various industrial applications. There are just a few examples of the industry where Saccharomyces cerevisiae is employed. Its versatility, adaptability, and well-established use make it valuable microorganism in multiple industry sectors. So next will be the usage of Saccharomyces cerevisiae in the industry. So firstly will be the alcoholic fermentation. Saccharomyces cerevisiae is responsible for primary fermentation in winemaking, where it converts the sugar present in grape juice into alcohol. During this process, the yeast produces carbon dioxide, heat, and various flavor compounds that contributes to the aroma and taste of the wine. Next will be the flavor department. Saccharomyces cerevisiae strain used in winemaking can influence the flavor profile of the wine. Different strains produce various levels of ester, alcohol, and other volatile compounds, which contribute to the characteristic flavor and aroma of different wine varieties. So next will be the malolactic fermentation. After alcoholic fermentation, some wine undergoes malolactic fermentation, a secondary fermentation process. In this step, certain strain of lactic acid bacteria convert to malic acid into lactic acid, resulting in a smoother taste and texture. Saccharomyces cerevisiae can help initiate malolactic fermentation by creating a favorable environment for the lactic acid bacteria. Next will be the sparkling wine production. In this production of sparkling wines such as champagne, Saccharomyces cerevisiae is used out the secondary fermentation in this bottle. This fermentation creates the bubbles by producing carbon dioxide under pressure. These strains with the specific characteristic are chosen to ensure consistent and desirable results. So next will be yeast nutrition and protection. Saccharomyces cerevisiae requires essential nutrients for optimal fermentation. Yeast nutrients additives are used to provide necessary nitrogen, vitamins and minerals, ensuring a healthy fermentation process. Moreover, these additives can help to protect the yeast from stress factors such as high alcohol levels and temperature fluctuations. Next will be the strain selective and improvement. The wine industry continuously explores and selects Saccharomyces cerevisiae strains with desirable attributes. These strains may be selected for their ability to ferment the specific sugars, produce desirable flavors, or withstand challenging environmental conditions. Genetic engineering and hybridization techniques are also employed to develop yeast strains with improved fermentation characteristics.
Metabolic process is utilized by Saccharomyces cerevisiae in the industry. It's commonly known as baker yeast. It is extensively utilized in various industries due to its metabolic capabilities. In the industry, Saccharomyces cerevisiae primarily undergoes a metabolic process called fermentation. Fermentation is an anaerobic metabolic pathway in which sugars are converted into alcohol, carbon dioxide, and energy in the form of adenosine triphosphate (ATP). Saccharomyces cerevisiae is particularly efficient in fermenting sugars, making it an ideal organism for industrial. In the food and beverage industry, Saccharomyces cerevisiae is used for production of alcoholic beverages such as beer and wine. During fermentation, the yeast consumes sugar derived from malted grains or fruit juice and converts them into ethanol, that is alcohol, and carbon dioxide. These metabolic processes are not produce desirable flavors or aromas but also contribute the carbonation of the beverages. Furthermore, Saccharomyces cerevisiae is employed in the baking industry. When it's used as the leavening agent in bread making, the yeast ferments the sugar present in the dough and releasing carbon dioxide gas that causes the dough to rise. These resulting gas bubbles give bread its soft and fluffy texture. Apart from the food and beverages sector, Saccharomyces cerevisiae is also utilized in the production of biofuels. The yeast can ferment sugar derived from various biomass sources such as corn, sugarcane, and cellulosic material to produce alcohol, which can be used as a renewable fuel source. In summary, Saccharomyces cerevisiae plays a crucial role in the industrial processes, primarily through the metabolic process of fermentation. It converts sugar into alcohol, carbon dioxide, and energy, making it valuable in the production of alcoholic beverages, bread, and biofuels.
In conclusion, Saccharomyces cerevisiae is a species of yeast that has been extensively studied and widely used in various fields, including biotechnology, food and beverage production, and scientific research. It is commonly known as baker's yeast or brewer's yeast. Saccharomyces cerevisiae has a unicellular structure and is classified as a eukaryotic microorganisms. It is particularly well known for its role in fermentation processes, converting sugars into alcohol and carbon dioxide through anaerobic respiration. This ability has made it crucial in the production of bread, beer, wine, and other alcoholic beverages. Understanding microbial growth and metabolism, including that of Saccharomyces cerevisiae, is of great importance for several reasons, which are industrial applications, food and beverage production, biotechnology and genetic engineering, disease research, and also environmental impact. For industrial applications, Saccharomyces cerevisiae is used in the production of a wide range of products, including biofuels, pharmaceuticals, enzymes, and organic acids. By understanding its growth and metabolism, scientists and engineers can optimize fermentation processes, improve yields, and develop new applications. Next, for food and beverage production, Saccharomyces cerevisiae plays a vital role in the production of fermented foods and beverages. Understanding its growth and metabolic pathways helps ensure consistent product quality, flavor development, and efficient fermentation processes. Biotechnology and genetic engineering. Saccharomyces cerevisiae is a well-established model organism for studying cellular processes and molecular biology. Researchers have extensively studied its genetics and metabolisms, making it an ideal candidate for genetic engineering and synthetic biology applications. By understanding its growth and metabolism, scientists can manipulate its genetic makeup to produce desired compounds such as biofuels or pharmaceuticals. Next, for disease research. Saccharomyces cerevisiae has provided valuable insights into fundamental cellular processes that are conserved across eukaryotes. Studying its growth and metabolism helps researchers understand basic biological mechanisms which can be applied to studying human diseases and developing potential therapies. Lastly, environmental impact. Understanding microbial growth and metabolism, including that of Saccharomyces cerevisiae, is relevant to environmental applications. Yeasts like Saccharomyces cerevisiae can contribute to waste treatment and bioremediation process by metabolizing organic compounds and reducing pollution. In summary, Saccharomyces cerevisiae is a versatile yeast species that has widespread applications in various industries. Understanding its growth and metabolism is crucial for optimizing industrial processes, improving food and beverage production, advancing biotechnology, studying diseases, and addressing environmental challenges. That's all from us. Thank you.